Well, let's explore the age of biology and chemistry in the O2H Innovation Conference. So I'd like to invite to the stage Tejas, who will be moderating your panel, and all the other panelists as well, Gayathri, Claudio, Sanjeev, and Anand Bhadalkar. Please give a big reception to this panel.
So we've been hearing the word biology, and I thought I would start out with something. If it squirms, it's biology. If it stinks, it's chemistry. If it doesn't work, it's physics. And if you can't understand it, it's mathematics. With that, we have an eminent and an august panel here. Thank you. And we have better representation from chemistry now. Great. Thank you, Andy. Right. So I have some bios here. Claudio, who's the business director at O2H, right? And before that, you studied for your PhD in the mechanisms of organic reactions at the University of La Sapienza, Italy. And you've been through multiple companies, academic drug developments, and other business development roles, and you end up here now. Would you like to tell us a little bit about your journey?
Absolutely. Thank you, Tejas. So I was born in Italy, and I've been interested, like Jeff, very early on in science, and specifically in chemistry. I didn't have a very good teacher, actually, so it was all my curiosity that made the journey start. So I had an opportunity after my PhD to come to UK because I was interested very much in medicinal chemistry, how to tune the structure of a molecule to make it active and make a drug. So I came to UK to work at GSK for 6 months, and this gave me the trigger to continue my career in medicinal chemistry. And at that point, I had the opportunity to come to Cambridge to work in different biotech companies. I don't know if you know Astex, fragment-based drug design, etc., and Celsom, proteomics. And after that, I thought that I wanted to do something else, and to do this transition, I had the opportunity to go back to university. I had a good contact with Professor Chris Abel, that was one of the co-founders of Astex. So he allowed me to go back to do research in fragment-based drug discovery applied to protein-protein interactions. And this opened another world, because the university gave me also the opportunity to co-found the Entrepreneurial Postdoc of Cambridge Society. That gave me like the opportunity to go out and see like a bit more in depth what entrepreneurship means and what innovation means. And finally, you know, I got into business development with Capital Cell, Abzina, for 3 years, like a biologics company. And at the end, Sunil Prashant asked me to join O2H, so I was quite happy to come and give my contribution in O2H.
So welcome to O2H.
Thanks.
And next we have Gayathri. Dr. Gayathri Sadasivam studied for her PhD in Switzerland, and she has a PhD in neuroscience. And from there, you have been through multiple drug discovery projects, companies, also some academia industry kind of collaborations on your journey here as the head of biology at O2H. So would you like to elaborate a little bit about that and tell us a little bit about your interests?
Thank you, Tejas, for the introduction. Yeah, I started my first scientific career, I can say, at the Indian Institute of Science, where I was working on my master thesis. So before that, I was fascinated by molecular biology, genetic engineering, but then I realized that's just a technology. It's not a big biological question. Then when I joined IISc, I happened to attend this talk by V.S. Ramachandran on phantom limbs. And I was really fascinated the way he cured phantom limbs just using a mirror box, and the way he explained synaptic plasticity, how you can use visualization as to bring in synaptic plasticity. And that fascinated me into neuroscience, and I wanted to understand the basic mechanisms of synaptogenesis. And that's how I started my career in science, doing a PhD at the Brain Research Institute, Zurich, looking at the molecular mechanisms of how synapses are formed, how they get adapted in the context of acetylcholine receptors.
Okay.
And then worked on dementia models, and later on moved into drug discovery, where I exposed myself to oncology, metabolic syndrome. And now at O2H, Sunil and Prashant have entrusted me a huge responsibility here in building the biology labs along with the UK and India team. And that's how I'm here finally. Thank you.
Welcome to O2H.
Thank you.
And we have with us Sanjeev. Dr. Sanjeev Upadhyay started out doing his PhD in the biophysics of ion channels at NCBS Bangalore. And then most recently, he is a UGC assistant professor at MSU Baroda. I will not elaborate a lot more other than to say he has some drugs in the market, but I'll let him elaborate about some of the interesting things he has done and what catches his interest nowadays.
So I studied biophysics, but as the fate has it, I picked up biology because I hated physics. And I ended up in a physics lab, biophysics lab. And my mentor said, here is a protein, you have to find out how it responds to voltage changes. So basically, whatever we feel are voltage changes which are propagated. And I said, look, Matthew, I'll take a little while to learn this. And he realized that he had made a mistake picking up a student. But while I was graduating, he said, no, I was not that bad, and you are doing okay now. And then I went on to learn how this knowledge of voltage potential changes can be used, and learned something about things which can block this, and then can we get something out of it. And it turns out that if you block one of these voltage changing machines we have, you can get thin. And I hope it gets into market. It is in phase 2 clinical trials. It is being taken up by a company called TechV in US. And then after finishing by those studies, once it is taken up by the company, you have essentially nothing to do. Then I picked up a job here in a pharma company, drug discovery, what O2H is doing essentially. 3 years into it, I realized that, I can do it faster than the company, a mistake. And then I picked up an academic job. Now the problem is how to find funding. And this is what I'm struggling with now. But I'm still working on the idea of drug discovery, small molecule, starting from the concepts of Ayurveda and trying to find some new molecules.
Welcome to the Innovation Conference. And on the note of funding, we go to the obvious source, Dr. Anand Bhadalkar. So, you are really close to scientists. You have been a scientist, but you're also detached from the field now. So what keeps you energized, and what are the most interesting things you're doing presently?
Thank you for that question. And yeah, I'm the oddest guy here in the panel. A passionate researcher for 10, 12 years before accidentally joining the government. I like the term biotechnology mission. I taught for 12, 13 years, and I was all drunk of APJ, who used to talk about mission mode programs. I saw this mission and I said, this is for me. And I jumped in. I got selected to realize that the mission was yet to be formulated. Okay, so I came in to बताया कि भाई table वेबल कुछ नहीं, आप बनाओ कुछ. But eventually, so GSBTM happened to be the first startup that we came up with. And all the learnings of this years of being a researcher and a faculty was now handy. I'd spent some time in CCMB and JNCSR, that was in ISC campus then in BRI. So we sort of developed 3 verticals, making sure that let every Sanjeev, wherever be he, be helped. Let every researcher be helped, let every student be helped, let every startup be helped, every MSME be helped. जहाँ जहाँ हमको लगा कि these are the things where government should contribute and work up. So that is how. In the meantime, I realized that I was bad with finance, so I tried picking up an MBA marketing and an MBA finance after that, and that sort of disenchanted me out of science, but still fascinated. But what fascinates me these days is making sure that we are able to deliver to the unreached the benefits of science and mechanisms, developing systems, making them sure that even after we move out, or the systems are in place to ensure that the delivery systems continue. That is what fascinates me these days.
Thank you. So on the point of delivery systems, I'll come to Andy, our CSO, and I don't have your bio, so would you just, for those of us who are new here, like me, tell us about your journey and about delivery systems at O2H?
No, my journey is very similar to as Jeff said. You have people who inspire you, and for me it was my chemistry teacher at school, who gave you the opportunity to be inquisitive, try things, and the thing that really decided me for chemistry was when I blew the school lab up, which was impressive, but it was the way then the teacher just kind of raised his eyes and looked at me and say, "What are you doing now?" But that gave me the kind of passion for chemistry and to want to study and understand it more. And then through university, it was trying to see that connection with the biology aspects and how those could be linked together. Okay. So my PhD was done in organic chemistry, similar to Claudio. After that, I wanted to do something a little bit different. And I went and studied in Japan with who is now a Nobel laureate in chemistry, because at the time I felt organometallic catalysis was going to be the future, and it has made a significant impact. All of you will be doing reactions of this type every day. From that then, it is looking at how you can utilize those aspects, and going into pharma was the kind of obvious choice for me, where you've got that opportunity to try and put those ideas, innovations in a way that, as Sunil talked about, there is potentially an end product that people out there, their lives can be impacted positively. So I had 20 years or more within pharma, and then I went to a conference, and this guy came up with a really off the way and different way of doing things, and that was Sunil. And straight away, you looked at this guy and thought, there's something about him, and it would be great if you got an opportunity to work for him. And that's where Opal, that Jeff's been talking about, kind of came on. And from that, we've kind of hit it off. We are very, very different. We've got our own strengths. We've got our areas that then you put them together. And my time then with O2H has kind of grown. And subsequently, I've been on this journey on version 2 with Sunil and Prashant for coming up for 10 years now. And I honestly have to say, this is the most interesting part of my career, and I'm looking forward to what's going to happen in the future.
Thank you. Claudio, on the point of organometallic catalysis, right? Asymmetric catalysis. This is what the Nobel Prize in Chemistry was awarded for last year. So, would you like to touch upon that and how that could affect, the kind of synthetic work that we do?
Sure. No, Tejas, you touched a very important part of the future way to make molecules. It's actually organocatalysis, not organometallic catalysis.
Okay, I stand corrected.
To understand this, we need to understand what are the two pillars, what were the two pillars of catalysis until like 20 years ago. One is the enzymatic catalysis, that with enzymes that allow two substrates to get closer together and to react in a way to lowering their energy of activation. So in biological reactions, so in our bodies. The other was the use of organometallics to make reactions happen, like reduction, etc. But there were lots of drawbacks to use these two ways of catalysis. One, for example, using organometallics is the high cost of the metal. They usually are transition metals like palladium, iridium, platinum, etc. And the other one, obviously, in enzymes is not so easy to make big quantities of them and to make it reliable to work in a scale-up reaction, for example. So someone 20 years ago started to think to organocatalysis. So instead of using an enzyme to make reaction happens, let's try to do it with small molecules. So molecules that have got like a weight of 300, 400, not like megadaltons. And basically this guy was Macmillan that won the Nobel Prize last year. Since the early 2000s, there was a lot of development on this kind of reactions. So now these molecules, organocatalysts, allow some, I'm speaking to the chemists here, enantioselective Michael reactions, Mannich reactions, hydrate transfer, even Mitsunobu reaction, enantioselective Mitsunobu reaction. And this allowed the synthesis and the scale-up of many, many drugs. I'm thinking about Rolipram, for example, or warfarin. They are all drugs but made in a different way, where basically we can scale up reactions without using organometal catalyst. It's expensive organometal catalyst and or enzymatic. And so it gave like a big input, a big improvement in the previous way we were making drugs.
Oh. So is that something that is already being used in pharma industry, or is that something that companies are migrating to now? What's your?
Yeah, companies are getting there now,
okay.
And in the last 10 years, let's say. So there were like more and more tuning and advancement on and expansion of the array of reaction that are already used in it. So definitely it's an important field to look at, especially if O2H will go into scale up. We will need to look at this kind of organocatalysis reactions. Okay.
So that is very useful. And then going from one Nobel Prize to another Nobel Prize in biology, we've all heard about Jennifer Doudna and Emmanuel Charpentier and the CRISPR-Cas9. So Gayathri, the question to you then is, would you enlighten our audience in relatively simple terms about what CRISPR-Cas9 is, and lead from that into, does it have an impact on drug discovery at all, or is it all academic purpose?
Okay, that's a good question. So I'll start off trying to explain in a simple way how CRISPR-Cas9 works. So essentially you have something called a guide RNA. So the guide RNA helps in precisely targeting, if you want to knock out a gene, it helps in precisely targeting your CRISPR-Cas9 technology to that part within the genome. So you have to use a programmable guide RNA in combination with Cas9. So now the question is, what is Cas9? Cas9 was originally formed in microbial systems as a part of innate immunity. So they exploited that as a part of this technology. So they combined the Cas9.
So this is something that is taken from bacteria and is now being
and
put
it is
in...
engineered.
Right.
So it is recombinant. So you continue to use the nuclease activity of the Cas9 and combine it with different types of guide RNA. So the guide RNA drives selective intervention within your genome, and the Cas9 allows you to cut within the genome. So in combination, it binds to specific sites, and then it creates something called double-strand DNA break. So once a double-strand DNA break is generated, there are two types of repair systems that happen within your body. So one is called the non-homologous end-joining pathway. This is highly error-prone and the most commonly occurring kind of repair pathway. So here you depend on this pathway for the reason that it's highly error-prone. It starts closing these double-strand DNA breaks by insertions and deletions, and they're called indels. And that's how mutations get accumulated, and essentially you are making the gene non-functional. The other repair pathway is the homologous recombination pathway. It's a high fidelity repair pathway, and it happens because it requires a hell lot of energy. It happens much more rarely. But then you use the homologous recombination pathway to knock in, specific sequences of interest. So both are exploited,
Okay.
and there are multiple applications. So the second part of your question is, what are the applications of CRISPR-Cas9?
Right.
So one of the most common application is, target validation. So what is target validation? So essentially, you have a clear clinical phenotype. You know what you want to look for. For instance, say, I just want to look at accumulation of lipid droplets as a part of obesity. I mean, I'm just hand-waving here.
Yep.
So that's the phenotype you want to look at. But you do not know what genes are actually causing this phenotype of lipid droplet accumulation. It's never one gene causing a disease. It's always a cluster of genes, right? So then you can use the CRISPR-Cas9 technology and do a whole genome screen to understand what are the cluster of genes that are leading to a particular phenotype or mechanisms, and then a larger phenotype. So you can map, as you can see. So that is one application. It essentially helps you in stratifying diseases. So how would you know what are the disease-causing genes? So you can perform genomics using this.
Right.
And the other application is transomics. So where you do not use the nuclease activity of Cas9, you engineer it in such a way that you fuse it to certain, you knock out, you just keep the targeting phenotype, guide RNA and Cas9, and you fuse it with transcriptional activators or repressors. So it essentially does not cause double-strand DNA break. It takes you to certain target sites, promoter regions within the genome that helps in transcriptional activation or repression.
Right.
And there are multiple things one can do. And even in generating animal models, for instance, it takes more than a year probably. You have to generate a single knockout, and then if you want to generate a double knockout, you should see if these breeds are pure with the back crossing and across the single knockouts, right? So that's not required. Already at a zygote stage, you can engineer, change multiple genes even before the development has initiated.
Okay.
So these are some of the applications, and of course there are many more.
There are many more.
Yeah.
So, have people understood this question, or are there questions about CRISPR-Cas9 and asymmetric organocatalysis that we would like to address before we move forward? No? Okay. So I'm just going to assume everyone is paying attention and has understood this. But on the topic of obesity, I'm going to come to Sanjeev because there was obesity mentioned, there was omics mentioned. I myself do not understand everything that omics encompasses. Would you enlighten us about, what does omics actually mean? Because what Gayathri spoke about is targeting specific genes, and that is one aspect of omics. But there is also this other, the big data aspect, right? So is that something that you would talk about?
So essentially, it is something that no human brain can comprehend. So suppose, for example, I wanted to know that I have a fatty liver. how do I go about it? One is the old way of taking my liver, slice it and look at it. The other thing is what else is going on besides having the lipid droplets? Or are there things which precede the formation of lipid droplets? So what I do is I take liver from people who are still thin but on the way to become fat, and we pick up their samples and look at everything which I can. So basically anything and everything which comes from these kind of livers, you look at it. So say for example, the common molecules we know is like glucose, cholesterol, phospholipids, and there can be more than 10,000, 12,000 molecules. If you study them by using modern techniques of mass spectrometry, you will find all of them, and then you try to see that what kind of molecules are generally seen in a young adult or a lean person or a marathon runner. Are there differences between these? So on a large scale, you have to use computer systems to actually look into all of these, and they will give you a bunch of metabolites, so to say, these small molecules, which are high or low in one person versus the other, or one group versus the other, or one race versus the other. And then you try to deduce that, look, okay, because of this whole bunch, we can now try to look for what enzymes could be more active here, or what transcription factors can be, or what are the drivers which can be up or low. Once we know this, we can look into the literature, what is known in science, and go back and ask this question, do they make sense? Many a time they don't make sense in the first go. But when you try to reiterate this process over and over again, 2, 3 different brains put together, they look into it, and then they say, here is some of the junk which you can keep aside, and the main data is these 5 molecules which have gone up or down. So this was about like metabolomics, what we call it, all metabolites to be looked at. You can apply the same thing to all kinds of RNA, the messengers which run from so-called nucleus to outside and give the message what to do, what not to do. You can look at all of these RNA and see what kind of messages are going more and what kind of messages are being suppressed. And looking at that, you can check, say for example, if you are working at a fatty liver person, that a fatty liver person usually sending more messages of this particular kind. Can we have a molecule which can block all these messages simultaneously so that we don't have a fatty liver? So there is actually a large data set from which we try to deduce some of the facts which we want to, and this gives us a good idea on an overall picture that what's going on in the system, because disease doesn't mean only lipid droplets, it also means sometimes a level of fibrosis, sometimes a level of insulin resistance, sometimes a level of enhanced glucose in your body, which is diabetes, and all of these put together. So can we look it in a holistic view that not one marker but several markers? For example, a person wearing a hat which is English style, generally you will say he's an Englishman, but he might be dark colored like me. So you look at 5 parameters, which tone he speaks into, whether he is speaking in a Hindi tone or a Malayali tone or something like this. You look at what kind of clothes he wears or what kind of food he eats. So if you have several of these parameters, then you are most reasonably be sure that exactly he or she is an English person or an Indian person. Or so similarly, these omics actually help us characterize one disease versus the other or one therapeutic versus the other in a more holistic fashion.
Thank you. I think that gives me a better perspective of what omics is. Andy, I'll come to you. These are things that are already happening. And from what I hear, Sanjeev, right, this is omics currently seems to be a lot of driven by the people, like manual analysis. Where next? What do you think? Where next do we go from this? What's coming next?
Well, we've talked an awful lot about tech, machine learning, AI, and I think in this first instance, lots and lots more data has been generated. There are algorithms to now start evaluating that. The power of computers has gone up dramatically. So there are these opportunities to do things differently. What are going to be the winners in those, Tejas, we will have to wait and see how things go. But it is moving things sometimes incrementally, sometimes significant step changes. And I mean, certainly as an organisation within O2H, we were very much to start with small molecule chemistry. Over time, things are now moving more towards opportunities where we're linking things with biology. So Claudio spent a lot of his career working on things like antibody-drug conjugates, which are a mixture of antibodies, the biological side of things, and then the small molecules, and using that in a directed and specific way in order to target diseases and get responses. And these kind of things at the interface, as we've been saying with the panel, omics, we're getting significantly more data. It's then how to mix and match that, utilize it, and using the powers of computers in order to do that, hopefully we can get there sooner, faster, and with better endpoints.
Great. So on that note, Claudio, do you have a take on AI, ML, harnessing the power of, harnessing computational power for what we do in a broader perspective?
Yes, I think it's like almost 20 years now that there are software that help generating new structure and new compounds in terms of, in a way to explore better the chemical space, because there are infinite possibilities to create a compound from a number of atoms together. So the problem so far was that these combinations weren't looking like a drug. They were just random compounds, and an experienced medicinal chemist should go through them and decide which were looking more as a drug and less as a drug. With artificial intelligence in the next few years, what we will see is that all this additional work from humans will be cut. And we hope that computers will generate more and more structures that look like a drug, that will address the problems of pharmacokinetics, that will be resistant to the microsome metabolism, and basically we will need to make less molecules to have results more quickly.
Okay. So I think that touches a very raw nerve for everyone who does drug discovery, because 90% of drugs fail before they can reach the patient, right? Gayathri, would you tell us why do so many molecules fail? Why do so many molecules, despite being positive as hits or leads, why are they not drugs? What happens in between?
So I think there can be multiple reasons for this, right from selecting the target itself and how it is getting translated from the in vitro to in vivo, the model systems that are used to validate, right, at the in vitro level. And then you're using a different species of cells, and then you're moving to in vivo models that are again different species. Although there is a lot of thoroughness that is tried, I mean, this is not intentional, but although there is a lot of thoroughness, how translation happens, that's another thing. And then once it reaches the clinic, the major issue is toxicity. The safety window is really low. Selectivity is an issue. So talking about the choice of target, because I've worked with undruggable class of targets, one thing that I have learned is you try to avoid targets that are on the surface of a cell, right? You're looking at cell surface receptors. And there is, you can imagine the number of pathways that bifurcates from the cell surface, right? Or you're looking at a kinase, correct? And a given kinase, or any kind of an enzyme for that matter, is capable of multiple post-translational modification. You can have acetylation, sumylation, methylation, phosphorylation. So a given enzyme is capable of making a specific modification on multiple proteins, correct? Right. So if I go with a GPCR, or for instance, a surface target or an enzyme, I'm essentially blocking the entire signaling bifurcation system, right? Yeah. So I think I'm having a very narrowed outlook here. The analogy here would be I'm cutting the trunk of a tree, okay, rather than cutting a branch. So these days, people go with something called post-translational modification. So if I have 20,000 to 23,000 genes, there are so many mRNAs being made, and there are probably a billion proteins, or million proteins, sorry. with post-translational modification. So if I want to specifically target and bring in precision medicine, I'll have to look at those targets that are downstream of an enzyme and at post-translational modification that are very specific and intervene between those two.
Right.
That's how I bring in selectivity.
Okay.
So that can be one of the answers, and I'm sure there are many more. I think that essentially takes care of these safety and toxicology aspects.
Right. So improving specificity is one of the things that.
Yes, definitely.
So Sanjeev, then the question comes to you: how, as biologists, right? What can we do other than what Gayathri mentioned about specificity? Because you have some experience with also, the drugs. What can we do in addition to this that would reduce failure rates or bring better drugs out? How do we balance the two without putting in too much more effort, right?
So essentially, Tejas is asking me how can you reduce failure. So there are two ways we can define this. One is that there is more toxicity and less efficacy in the later part of research, and how can we reduce this? Part of it is because of the technical issues which are involved. But what I personally feel is a major reason is that we are not honest as a researcher, as a company running person. So here is the thing, what I think personally, may be politically incorrect. So there are two kinds of people involved in making a drug. One is the scientist and one is a businessman. Businessman actually wants to make money. That is his primary goal. And scientist actually wants to solve a problem, but within last few years what has happened is scientists are more into publishing paper rather than solving problem. So research study about almost a decade ago, Nature Publishing Group had established a lab and looked at some of the more impactful papers in recent history and tried to reproduce those data in their own lab. More than 50% of data was not reproducible. So here is the thing, if I work on a target or in a validation, and if that is something which I have not worked personally, I'll not pick that target. Because somebody else has done it to publish a paper to get a grant, and if I invest $1 million and fail, that's a risky line to take. So for me, it's about the human nature which is causing this more failure. We have less and less drugs coming to the market. For example, I was working in a CRO company some time ago before joining academics, and we had a target to work on, and it was a lead optimization stage. And many of you who work this would understand what is lead optimization. Means most of the work has already been done, so to say. And 3 months into it, and I said, it is going nowhere. This is a pathway you are targeting is wrong. I mean, it will lead to the toxicity. No, this is molecule specific. Why was this? The person driving wanted to keep his job, so he invested more money. Therefore, in academia, we keep publishing a theory which we partially know may not be holding true. We ignore some of the data which our students present to us. No, this doesn't fit into my hypothesis. Just put it on the side. So both ways create a huge failure later on. And a typical drug cost, which was less than few $100 billion about 20 years ago, is now $2.6 billion. This is because of these failures. So this is my take on failures in drug discovery.
This is a very important point. That the honesty in this answer is something that deserves this applause. Thank you, Sanjeev. And which also brings us to the culture at O2H, where you're not blamed for your mistakes so long as you own up to them, I guess. And but you're also, asked to take risks, asked to take charge of things. And the point that Jeff made about doing good science, trying to put your best foot forward, doing the best that you can, so that the person at the other end knows you have done your job honestly, and this is the best that you can get, right? And so thank you so much for bringing that point forward. And I'll move to a slightly different question with Anand. Where next? So we've been talking a little bit about drug discovery. We've heard about investment in biotech space in general, which also spoke a little bit about drug discovery. To 20 years down the line, will drug discovery still be the thing, or are there other things? Like green tech was a word that was mentioned, right? So what is your vision?
Before I take this question, we have been talking about a couple of things. Say, for example, we talked about obesity. So in government, state government, and central governments have started believing that yes, biotech has solutions for most of the things. May it be obesity, may it be malnutrition. If you're talking only about food, malnutrition, nutritional adequacy, obesity, we have solution out there. Swachh Bharat, bioremediation, we have solutions out there. Hence, biotechnology is the ray of hope now, and that is where next investments, public investments should come in. So when we look at how have we tried addressing all of this, okay? So culture, as very correctly mentioned. So we tried that, yes, let us support researchers so that they build their, state researchers have not been able to make it from the national funding and so on. So let us build their CVs, let us give some state-based support systems, okay, so that they build their CVs. Now they are able to get SCRB, DBT, DST funds, okay. So we are a funding agency for research ourselves. We have been funding startups and MSMEs, okay. And Prashant was there when we had a launch of the new policy very recently, where we have not gone for an incremental rise, but for a quantum leap, okay. And we had two new policies that came in, the biotech policy and the IT policy that came in just before that, because we talked about AI, ML as well. So I'll just try and quickly mention that, wherein we have gone for a quantum leap from पहले जो है per unit 5 crore, 10 crore, 25 crore के आसपास देते थे, okay. From there in IT policy we have gone to something as high as 600 crores per unit, okay. From in biotech we have gone to something like 450 crores, okay, per unit. Now this, it talks about the resolve of the state government to come and support. Now this is grant support, unlike equity, okay. This is grant support. पाछा देवाना नथी, okay. So this is the government's inclination towards supporting. Apart from this, building capacities of students, researchers, entrepreneurs, clinicians, doctors, lawyers, farmers. So we have a separate vertical which is working towards this. Having said this, now I'll quickly tell you, especially drug discovery or so on. So building other infrastructure, we did talk about in the previous panel, where some of you would have heard that, wherein the state government is developing incubators, parks, okay. In addition to this, also some very interesting places like the Gujarat Biotechnology Research Center, okay, with shared facilities, which means that any researcher out there, any student wanting to use an equipment, come use it. For students and startups, it is at subsidized rate. For industry, it is at slightly higher rate. I mean, for academicians like him, it is at the cost rate, okay. Just come up only with cost of consumables and it's done. For industry, it is 1.5 times of that. But these inclusive of sophisticated instrumentation is available. You want to get into drug discovery, we help you with instrumentation, with funding from GSBTM, with this. In addition to this, the state government has openly come up next to the biotechnology university that the state government is developing with University of Edinburgh, okay, coming up with a preclinical testing facilities inclusive of animal testing and so on and so forth at the cost of 250 crores, okay. BSL-4 facility. Now, you can imagine the kind of impetus it will give, okay. We talked about AI, ML, okay. Everyone now is clear that we need to pick up some courses onto AI/ML, we want to pick up some courses onto omics, we want to pick up some courses, okay. So the IT policy allows you to, we'll list out some number of these courses, but you pick up these ones, 50% of the, 50% of the course fee will be borne in by the state government, okay. State government is coming up on the lines of the biotech university and AI university, which will be a first of its kind in Gujarat, okay, using state government funds, university dedicated to AI/ML, because next 10 years, as you said, are going to be extremely interesting, and we have things all consolidating right from across technologies to things like this, and hence, yes.
Okay. So I'll use that same question, pass it on to Andy. Andy, as the CSO, where next? What's your projection for chemistry, for biology, for O2H? Where do we go from here?
I think we're only on the tip of the iceberg. There is still so much more to discover, understand, and move forward. Science always evolves. So where we were 10 years ago, huge things have become the norm. And they are going to be things that we just do not envisage at this moment in time. I wish I could be the one with the crystal ball, if it was like Jeff with understanding what the pandemic's going to do. If we knew that, we'd all be rich, we could retire and everything already. So it is going to be those kind of eureka moments. And it is getting that into the process. You talk about O2H, the culture, or in general, we have to be efficient in what we do, and that is processes, but at the same time, rigid processes don't allow innovation. And it's how we get that balance, how we can use all the skills and capabilities that are around us to allow us then to get to where we need to go. And I think there is an awful lot of stuff that we haven't even tried and looked at. Hopefully the interface between chemistry and biology are things that haven't been explored. We are starting to do that in certain ways, but there's going to be a whole load of therapeutic opportunities that will present themselves in the future that are, at this moment in time, kind of mind-blowing, because we'll look at them then and go, yeah, that is wow factor. Okay. But we just don't know where they are at this moment in time. So I'm sorry I can't give you an answer.
No, I think that is that's a good seed. That's a thought seed, which allows all of us to think more critically about what we see, what we hear, and what we read. And what does the audience think? Do you have any questions about the discussion that we had? Do you have different questions? 2 questions down there. Perfect. Mike, please.
I have this question about an example that I have seen recently. We saw the fastest FDA approval in history, I think, for the COVID vaccine, right? So for the drug discovery or integrated biology and chemistry discovery, how do we expedite that process in general rather than in emergencies, rather than just in the pandemics? I wanted to know your take on this. Like, do you think that FDA would be much more faster in approving drugs from here on? Thank you.
Gayathri, I'll direct that question at you.
So I think the speed of drug discovery has definitely improved compared to 10 to 15 years. Now there are multiple approaches. One is a target-based approach. So in the case of pandemic, there was a dearth need. So, I mean, everybody put forth their best, forgetting all the geographical boundaries, and there was exchange of knowledge, exchange of publications, and it's a crisis situation. The analogy to this is siblings can have a lot of fights, but when there is a real crisis, they come together and combat a difficult situation. But then you're asking, how would this pan out in a general situation, right? So one is a target-based approach, then there is an omics-based approach, and then you have a hell lot of data, information available, right? Algorithms that are available. Everything is just algorithm, right from bacterial system to, higher eukaryotes. Now, even with the whole genome sequencing, you have like about thousands of genomes being sequenced just for cancer. It's a humongous information. Now, how do you process this information is something that we've been talking about, AI and ML. So it definitely needs an integrated approach. There's a top-down approach and a bottom-up approach, target-based way of identifying drugs, and then there's a phenotype that you have in mind, and then you deconvolute what the target is, and then you use AI and machine learning to understand and map at a genetic level, then at a pathway level, then at a clinical outcome level, look at robust biomarkers that can be modifiable. And using this information, then you come up with new drugs. So you have a repertoire of drugs against a repertoire of disease types, disease stratification, so that the mapping is accurate. Otherwise, you cannot move forward, and definitely it's an integrated approach that can take drug discovery forward faster than what it is. Hopefully within 5 years, I hope so.
Right, so
And
I think that was...
Just to add something to
I'll
your
just
answer.
add saying that
Gayathri, basically, really is to shorten the approval process, the FDA approval process. The main thing would be really to look at a really good improvement towards like the current medicines. So if you have got like a sort of improved effect or a very clear effect of these medicines, the clinical trials, the approval will become really much faster.
Okay.
Instead, if the effect is not clear, which is always very often is the case, then the approval will be much slower. This is like, from the approval side of the things. Obviously, from the research point of view, we need
There's
to have like
another
this-
question,
in addition
Rupal
to
or
hoping
Sinush?
for it, we'll need to work for it. Okay, we'll need to push things.
We'll just take a question.
hoping
I think
along.
we have a question there.
Hello everyone. First of all, thank you for such an engaging discussion. This is for, I think, Gayathri mainly. So we spoke about CRISPR, and speaking of advancement of technology, recently at Harvard, a group tried something called programmable base editing, where they were just targeting nucleotides, changing A to T or G to C. And that's something that was mainly for single point mutations, for example, cystic fibrosis. So my question is, what is the promise that you see in such advancement in technology over, let's say, CRISPR in this case, and how does it impact drug discovery or therapeutics for diseases? Thank you.
Thank you, Sinosha. So that's one good example that you have given, other than, we were talking about disease stratification, then we were talking about transformics, where I gave you an example of fusing the Cas9 with activators or repressors, and one example that you just given is, base transitions. Drug discovery, one other application, we are always talking in the context of humans. So there is another thing called gene drive. So that is a very, CRISPR-Cas9 also falls into picture there. So for example, if you're wanting to eradicate tropical diseases like malaria, so you can use gene drive. So the way, for people who do not understand how this works, is so if you have a mutation, right? So the way it propagates across population is using Mendelian inheritance, right? And with time, this mutation is diluted and it dies out. It's as simple as saying your father has blue eyes, your mother has, brown eyes, and then you see this blue eyes popping in time to time in the following generation, but then it completely disappears. So that's because of the natural evolutionary selection or choices it makes. But in the case of gene drive, the way they engineer CRISPR-Cas9 is that they use the sequence of interest, and then they have Cas9 engineered, and then they have clusters of guide RNA. This is engineered within one of the chromosomes, and the other one is wild type. So the male, for instance, this is in a male mosquito, it crosses with a female, and the next generation, all of them are heterozygotes. So when you have heterozygotes, again, mate, there is a 100% chance of within the second or third generation, they carry this gene of interest. So you can see how powerful this is. And I do not want to eradicate mosquitoes. That can be very dangerous. So ideally, you keep the mosquito population, and probably you bring in sterility, right? That's one way of removing malaria. It sounds like science fiction, but actually it is happening, and this can be adapted to many other scenarios. So if you read Yuval Noah Harari's thought process, the most powerful thing in today's hand is information technology, and the other one is biotechnology, and it can do wonders. It can destroy or it can create.
And with that, we'll take the last question from Prashant. Oh, is there, I thought you had a question, but there is a question in the center here.
Hello. Yeah, Tejas, you already touched the failure of drugs, but my additional question is that why Indian discovery is a failure in NC molecule point of view? This question to Gayathri or Andy.
Andy, do you want to take that? Could you repeat your question? Why is Indian discovery?
Indian discovery NC molecules failure more than Western countries.
Why is NCE discovery delayed?
It's probably not down to me to comment on that. I mean, from a cultural perspective and everything I see here, you've got all the skills to be able to deliver it. Whether it's governmental, whether it's financial, or India's such a strong tradition in manufacturing, it's just carried on that way. And somehow the cycle needs to be broken.
Okay.
I don't know. But you've got all the skill sets and you've got all the drive and innovation opportunities to do it. I see it every day with my colleagues. So why that doesn't translate, I don't know.
Okay. I think that's a very hopeful answer. And on the note that we can literally play God with biotechnology, we can innovate with NCEs, chemistry, and there is a lot of hope for the future, not just for drug discovery, but for a lot of other things. And I'll just thank all the panelists here for their contributions, for educating all of us, and for coming here. Thank you.